started. So when I was going over the PowerPoints today, I figured either today or Wednesday, we wouldn't be able to clear everything that I had in store. So I did the thing of where I moved back all the folders a day starting on Wednesday. So Wednesday, there will be no reading guide due, and that's already been changed up on Blackboard. So no reading guide on Wednesday. Friday is our exam. The material we cover today will be on the exam, the last amount of material for the exam. What we cover on Wednesday will not be on the exam though. So today's material will be the end of it. So let me pull up that PowerPoint. So what we were talking about on Friday is we started looking at the lipid membrane, right? So we talked about the FRAP experiment, F-R-A-P, FRAP, which is shooting a fluorophore with a laser, kills that fluorophore, and then you can see how long it takes for something to come back into the spot that you shot. Then we talked about an experiment in which we saw that proteins in the membrane are mobile, where you take a mouse and a human cell and you combine them together. Human proteins are red, mouse are green, excuse me, mouse are green. And you just look at the fluorescence, and over time you'll see that the red and the green overlap, which means that these proteins are moving. And these types of experiments allow us, or have allowed us to come up with a model of the membrane, and it's a fluid mosaic model. Fluid in that the things inside your membrane can move. Mosaic in that it's made out of a lot of different things, as shown in the picture here. You have your cholesterol, you have your lipids, you have integral proteins, you have lipid-linked proteins, you have carbohydrates, glycolipids, all of that. Then the last thing we talked about on Friday was Spectrin. Spectrin is the protein that makes up the skeleton of the membrane. It can be connected to ankyrin right here. And ankyrin is connected to an anion channel. And this just says glycophorin, which is another protein, right? And by understanding that spectrin is part of the skeleton, we can start to understand some of the various movements of proteins we see in the membrane, which is shown here for A, B, and C. So this is a gate and fence model of the membrane. And what that means is that some proteins, like protein C, has free movement. And this green is supposed to be spectrin. And we'll see that if you put a fluorophore on C, you'll just see it move all over the place. For protein B, that has movement in one area of the cell because it's in this gate. And we'll see that when you do protein B, put a fluorophore on it, you'll see that it'll move, and sometimes it'll move out of that area, but it'll be stuck in one area. And then protein A, where this protein's actually on the cytoskeleton, or it's not the cytoskeleton, but the membrane skeleton, and it can't move at all because it's interacting with spectrin. All right, so spectrin allows us to understand the different types of movements we see for proteins in membranes. So what we're going to start is just some questions to make sure we understand those previous slides. And so I have a poll for a lot of these questions. So let me pull that up. And the poll should have, yeah. So here we go. So take a minute or so and see if you can answer this poll question. All right, seems we have everyone pretty much voted. So let's take a look at this. So what experiment does not really verify the fluid mosaic model of membranes? So the correct answer here is B, right? So A, let's just talk about why A, C, and D are. So A, you have a cell and it's stuck on a microscope slide. Why that is important is that if I want to see a protein moving in the membrane, the cell can't be moving, or else you can make the argument, well, it's not the protein that's moving, it's the cell. So you stop the cell from moving, and then you can put like a fluorophore on a protein, and if you see it moving inside the membrane, that tells you, your membrane's fluid. C is what we just talked about at the very beginning here. You take that human and you take that mouse, make it into one, and you'll just get human and mouse proteins moving all over the place. And D is more or less the same idea as an A, where you take your protein, put fluorophores on it, shoot it with a laser, you bleach it, and then you see if the fluorescence comes back. So D is FRAP. And this will show that your membrane is fluid. B, where you take a human protein and you put it in a mouse cell membrane, and you just see transverse diffusion. Transverse diffusion is going, right? So lateral diffusion more or less means going from like left to right. Transverse diffusion would be going from one side of the cell membrane to another. So one, that doesn't really tell you that your membrane's fluid. That's just meaning your protein can flip back and forth. One, that would be really hard to measure. But also, that's not really an experiment that was done, because an argument you can make for that is, well, you're introducing a protein that's not native to the membrane. So that's why we're seeing movement here, is because you introduced this whole new protein. So B is not really a good experiment that will say we have a fluid mosaic model of the membrane. Questions about that or that explanation? All right, so we'll move on. Question 2. So we talked about different proteins on like 2 slides ago where we're looking at the Gates and Fence model. I'll just redraw it here super quick. Something like this, where you have like protein A. Let me get a new color of pen. Protein A being stuck, protein B being in a little pen, and protein C able to go everywhere. So why are some proteins like protein A stuck at a certain part while something like protein C can go anywhere in the membrane? What would be possible reasons for that? Why do you think that happens? Well, the Gates and Fence model is just saying that some proteins are gated and some proteins like jump over a fence. So that's just saying what the model is. Why do some proteins just have to stay at one position? Maybe its function only works on the part of the cell's anchor to. So that's kind of what I was thinking and what I was getting at, right? So some proteins in the membrane are just stuck in one place of the membrane. And so the question is, what advantage is there for a protein to be at one part of the membrane only, to give it no movement? And one of the obvious things or one of the obvious answers I would think is that, well, it has to be there to do its function, right? So one reason why you would make a protein like protein A just be stuck at one location, because it needs to be there to function, right? So that's what I would think with that. Needs to be in a certain location to function. And since it was asked, I'll just reiterate what the Gates and Fence model means. And let me go back 2 slides for that, just so we're all clear. Whoops. When I was saying Gates and Fence model, right? That's this. And what this tells us, what this actually is showing, and let me do it, is that when we look at proteins, protein diffusion in the membrane, what you'll see is that, and this thing I'm drawing is like protein movement in the membrane. You'll see it stay for a long time in one area, and then it will like move very far, very fast, and then be in another area, stuck in another area. And then after a while, it will move to a different area and be stuck there for a while. So what that is showing us, like if this is diffusion of a certain protein, and those 3 colors are just different time points, is kind of what proteins B and C are showing us, where here we kind of get stuck in our spectrin like fence, and we're stuck there for a long time. And sometimes we can find our way out of that fence because there's an open gate and go to a new area and be stuck in a new area. So that's what our different time points are showing. Sometimes, like protein A, we just get stuck in one area. So protein A's diffusion would just never move because it's actually on the spectrin membrane. Are there more reasons? That's the only one I could think of. If you have other reasons, I would love to hear them. I'm not saying that's the only reason. It's the only one I could think of. So any questions about our spectrum model or anything so far? All right. So we talked about this a little bit in a previous lecture. I actually had a question on this idea in a previous lecture. But when you look at the lipid bilayer, the two sides are not the same at all. The two sides are quite different, right? And by the two sides, you usually call them the inner leaflet or the outer leaflet or the cytoplasmic side and the extracellular leaflet or side. Right. So the leaflet, when you see leaflet, when they're talking about a bilayer, they're just talking about one side of the layer, right? So they're not uniform at all. And we can tell that here on the bottom left, we see 4 different types of lipids. So we have sphingomyelin, phosphatidylcholine, phosphatidylnethanolamine, and phosphatidylserine. And there's different amounts on each side of the leaflet. For example, phosphatidylserine is only ever found in the inner leaflet. It's never going to be found on the outer leaflet. Now, to make these lipid membranes, or these lipids, the enzymes in eukaryotes are found in ER, and they're integral membrane proteins. And so just as a reminder, an integral membrane protein is a protein that goes all the way through the membrane. So that's where our lipids are made. Prokaryotes, they don't have an ER, right? They don't have organelles. So they make their stuff by integral membrane proteins in the plasma membrane. Now, you might be asking yourself, how do we get this big difference in lipids? Well, we have some enzymes that specialize in moving lipids from one leaflet to another. So one is called flipase, and this is the flip-flop of specific phospholipids. It's facilitated diffusion, where it goes by a concentration gradients, right? Where we have a lot of something on one side, so we can flip it on the other side. Not listed here, but there is another protein called floppase that works just like flipase, but it goes in the other direction. So flipase goes one direction, floppase goes another. And there's also something called scramblase that flips 2 things at once. So the people who came up with these names, not very creative, but they're telling you what's going on. Flipase, floppase, scramblase. You also have specific phospholipid translocases. So all they do is using ATP, they will make phospholipid go from one side to another. So for example, let's say we are making phosphatidylcholine and we have it on the inner leaflet and we want more of it in the outer leaflet. You're probably going to have to use ATP to flip it to the outer leaflet because it's higher concentration. You're going against the concentration gradient when you do this, so you probably need ATP. Now, depending what type of lipids you have on the outer leaflet, can be used as a signal for, and this was the question I actually asked a couple lectures ago. So phosphatidylserine, usually only found in the inner leaflet. So if it's found on the outer leaflet, that means a problem has happened. And one way that you start the signaling process for blood clotting is if there's phosphatidylserine floating around. Because if there's phosphatidylserine either showing or floating around in the bloodstream, that means your cell has been disrupted, which means you're probably bleeding somewhere, which means you need to seal up the wound. So that is one way blood clotting can be started. Flipase, floppase, scramblase, yeah. I mean, they tell you exactly what they're doing, yeah. All right, any questions about the information presented here? Myself, I like when people give, I guess I don't like it, but I always find it funny when people give proteins stupid names like Sonic the Hedgehog. and Sonic the Hedgehog is inhibited by Robotnik. You can't name proteins like that anymore because Sonic the Hedgehog causes cancer, and doctors were feeling really bad about telling patients, yeah, so your Sonic the Hedgehog is mutated and you have terminal cancer because of it. So you can no longer name proteins anything you want, unfortunately, because of Sonic the Hedgehog. All right, so that's my fun little diversion into protein names. All right, here's another question. And I have a poll and it's basically true or false. Can we use FRAP to measure the following? So let me get that poll up. Or I guess I have it useful or not useful. So for all these questions, tell me, can we use FRAP for these? Why do they need a form of active diffusion when they have all the forms of passive diffusion? Because sometimes if you're going against a concentration gradient, you can't do passive diffusion. Going from low to high concentration takes energy because you're going against entropy. So that's one reason why you need active transport when you are trying to go from low to high. I mean, so like the example I can think of for biochemistry that you might have taken in a class already is the electron transport chain. The whole reason you do the electron transport chain is because you are trying to pump hydrogens on the outside where they're already high. And so to do that, you need energy, and we get energy through the way of electrons, not ATP there. Then at the very end, we let hydrogens go from high to low naturally, and we use that energy to make ATP. If we ran in reverse, you can spend ATP to pump it back out. So this question is a good review of, do you actually know what FRAP is? And just as a reminder, FRAP can stand for fluorescence recovery after photobleaching. Or another name is Forster, I think probably named after the guy who first proposed the mathematical relevance of this. So we'll give, I don't know, about 20 more seconds for this. All right, let's share this. So before I answer, let me just briefly go over FRAP again. So you have a cell, and inside the cell, you have proteins with fluorescence. Actually, let me just color it in. That's going to be faster. You shoot the cell with a laser at a certain spot, and that makes the fluorescence go away because your whoa, I erased everything. Undo, please, because that, so you make one spot go away because those fluorophores get quenched. You basically destroy them. And then you see if your proteins can go back in or not. So can you use this to measure the concentration of protein in a cell? You cannot. You can use fluorescence to do that, sure, where you can see how much fluorescence you have. Based on the intensity of the signal, you can start to guess like the amount of protein. It's not a great way to measure protein, but it's a way to get like a sense of protein, right? But FREP, there's no reason to shoot your proteins with a laser if you're measuring concentration. So the actual FREP is shooting it with a laser. So that's not useful. Diffusion of a protein in a cell, well, that's very useful, right? Because you are taking one area, making sure that any protein was there, the fluorophore is destroyed, and then you're seeing how fast does it take to refill in that area. Using some calculations, you can determine inside the cell how fast does my protein move. And that's one of the principal uses of FRAP. The speed of flip base, oh, not useful. Remember what flip base does, right? We have a lipid bilayer, right? So here's my bilayer. Flip base takes one lipid and flips it to the other side. And you are looking at this at a microscope, right? There's no way you could ever tell what side of the lipid or what side of the membrane layer your lipid is on by looking at it through a microscope, because your cell looks like my drawing there. It's a flat circle, right? We do not have microscopes that are good enough to look at which side of a bilayer lipid is on. So this is not useful for measuring the speed of flipping lipids from one side or the other. However, you can put a fluorophore on a lipid and just see how fast it goes inside the membrane. That's perfectly fine. But we don't have tools that are good enough to see how fast it goes from one side to the other using FRAP at least. You can do that through other calculations, but not FRAP. Any questions about that? You like these questions? Well, hopefully you like them when you see similar stuff on Friday. Why is D useful? Because lateral diffusion. So here's a cell, right? Lateral diffusion inside is like moving across the lipid membrane. And so it's the same idea as an A that I drew here. Let's say you have a specific lipid and you make a fluorescent version of it. I'm not saying that's easy or even possible for every lipid, but some lipids are fluorescent. And so you just see how fast does my lipid move in the bilayer, right? So you put your fluorescent lipid in the bilayer and you let it equilibrate. So there's fluorescence all over the bilayer. You shoot a part of the bilayer or your cell with a laser, right? And then you see how fast it takes to refill in. And from there, it's an easy enough calculation to see how fast do lipids move in the bilayer, which will tell you how fluid your bilayer is, which, so on and so forth, leads us to the fluid mosaic model of the bilayer. So it's experiments like these that we can really understand cell biology. How does cell biology work? What else would FRAP be useful for that's not listed here? FRAP itself, I cannot think of like a particular good reason other than cell movement or protein movement. There's a lot of stuff you can do with fluorescence that does not use any lasers. But FRAP is usually to see if something is mobile or the speed of mobility that I know of. So usually it is these things. All right, because you give everything a fluorophore, You kill some of those fluorophores, and then you ask, okay, can I now move? Or is everything being created and just stuck at one place? Because if you don't see anything filling in, that tells you your protein is forever stuck at that one place. It will never move. Or it can tell you like this protein's super slow or this protein's super fast. But yeah, you can do a lot of other stuff with fluorescence. Unfortunately, we're probably not going to talk about all those cool things. If there was a biophysical class, we definitely would, but there's not. All right. So let's continue to talk about the lipid bilayer here. And when we study the lipid bilayer, we'll see that when people have studied, they saw that there are areas of organization. And one of those areas is called a lipid raft. And basically a lipid, let me rewrite all the words here. So what's being shown here, these green things are cholesterol. These blue things are transmembrane proteins. And this is glycosphingolipids. And what a lipid raft is, it's just an area of your membrane that is more organized. Where here we have a lot of protein and cholesterol and glycosphingolipids. And really, when you look at the plasma membrane, this is why it's called a mosaic. You'll have different areas with different domains. That is, different areas of your membrane will have different lipids or different proteins. So totally get out of your head this idea that the lipid membrane is just like one uniform thing of lipids. It is much more complex, and it is very different depending on where you look at the membrane, right? And so a lipid raft is just a micro domain. It's like a small little area that has a lot of glycosphingolipids and cholesterol, and it's not that fluid compared to the rest of the membrane. That's why it's called a raft. This area is like a solid that floats in your lipid sea, right? So think of it literally as a raft, and think of the lipids as the water in which it floats in, right? And so different proteins will associate with these rafts. So your GPI proteins, you remember the GPI link proteins are the proteins with those the big sugar head, and that's how it attaches to the membrane. They're looking for these lipid rafts. This is also thought to be important in cell-to-cell signaling. These little platforms can be like stations where we put proteins that are necessary in cell-to-cell signaling. And also, lipid rafts are one of the ways in which your cells become invaded. So the flu, measles, Ebola, HIV, the initial way they get into our cells is that they attach these rafts. They use these rafts as their staging point to invade the cell. So any questions about our lipid rafts here? Right. So question 4, going back to red blood cells. The shape, the biconcave shape, and what they mean by that is like the donut shape, right? So red blood cells basically look like a donut. That's a horrible drawing, but that's a very bad drawing, but it's like a donut shape. So the biconcave shape of erythrocytes, which maximize rapid diffusion of oxygen, is probably maintained by, let me get my pull up. So here's another refresher question for us to make sure we've been paying attention. So let's see if we can get this one. And you might see questions like this coming to you this Friday. do about another like 30 seconds for this one. All right. So the correct answer here is C. So remember, spectrin is our membrane skeleton, and the red blood cells have this very unique shape to it, where you have like this ditch that goes in the center of the red blood cell. And that is solely due, or mainly due, to spectrin, where you just have a ton of spectrin on the outside of your red blood cell that's conferring this really unique shape to the cell. So the answer to that is C, spectrin, which most of us got. So that's a good little refresher from Friday for us. And I have another question for. So let's see if you remember what I said earlier. So I have two different concentrations of lipid. One of them's inner leaflet, one of them's outer leaflet. You tell me which one's which just by looking at what's in there. So I'm telling you, just by looking at the lipid composition, you can tell me if we're looking at an inner leaflet or an outer leaflet. See if you can figure out how to do that. And we did talk about this today already. All right, so let's do 10 more seconds, make an even 2 minutes. All right, so the top one is inner, the bottom one is outer. How to figure that out? Phosphatidylserine is a lipid that is only in the inner leaflet of eukaryotes. If phosphatidylserine's ever on the outer leaflet, that's a signal for something's gone wrong. So phosphatidylserine showing on the outside is one way for white blood cells to come and eat that cell. And it will also start blood clotting. So if you ever see phosphatidylserine, unless something's gone wrong, you know you're looking at the inner leaflet. Questions about that? No, it's pretty straightforward, but. No, we will move on. All right. Now, any protein that needs to be exported out of the cell or is an integral membrane protein, so Oh, I do have some questions here. Wait, what about the outer? How can we interpret the other one to know it's the outside if it doesn't have phosphatidylserine in it? Right? If it has no phosphatidylserine, it must be outside. If it does, it must be inside. Yeah. What if they both have it? You're looking at a cell that's about to die, right? If we go back to this chart, phosphatidylserine is only shown on the inside. So everything else is more or less in both. So if phosphatidylserine is on the inside, that's or if you see phosphatidylserine, that's inner leaflet. If you don't, that's outer leaflet. Phosphatidylserine is always inner. Yes. Always, always, always. Like I said, if it's on the outside, that either means the white blood cell is going to go and eat that cell very shortly because it is dead or is dying. Or it can also start the blood clotting process because that means you have just ripped open some cells, which means you're probably bleeding. So yeah, phosphatidylserine is always on the inside. All right. So getting back to this pathway, would you ever give us anything but phosphatidylserine and have us find out what's on the inside and what's on the outside? No. I promise you I will never ask that question. All right. So the secretory pathway. That being said, I probably won't ask you that same exact question again because we just went over it, but you never know. I might. We'll see how tricky I'm feeling when I write the test on Wednesday or Tuesday. Okay, the secretory pathway. So any protein that needs to be transported out of the cell or is an integral membrane protein that must go through the ER before it is ready to go. So the question is, and this is roughly, 25% of all your proteins are either going to be transported out of the cell, and it can range up to like 40% depending on what cell you're talking about. So these need to go to the ER. So the question is, how does the cell know what proteins need to go to the ER and which ones don't? And that's all due to this pathway. This pathway is showing us how it's done. So we start with how to recognize if a protein needs to go to the ER. And it's through an N-terminus tail. Not tail, but signal, sorry. N-terminus signal. Remember, the N-terminus comes out first. And what you're looking for, and it varies a lot, is that you're going to have some positive amino acid. And then you have a bunch of nonpolar amino acids following that. And so this, if we line up the signals, and I'll erase all that so you can see it better. If we line up the signals, you see it doesn't overlap that great, but we do have that pattern where we have some positive amino acid and then a lot of hydrophobic amino acids. So this is a signal. And what happens when your protein is growing on the ribosome? You have a protein called SRP. Signal recognition peptide. Signal recognition peptide. And what this will do is that it will have GDP originally bound to it. And it will go and it will bind the peptide. When it binds the peptide, GDP leaves and GTP comes in. When GTP comes in, SRP goes through a conformational change. And that conformational change stops the protein from being made because this protein needs to be modified in the ER. So it cannot be made in the cytoplasm. So SRP makes sure this doesn't happen. Once the peptide has been stopped, SRP will go to the SRP receptor on the rough endoplasmic reticulum membrane. So that's what we're looking at here. That's what this gray thing is our rough endoplasmic reticulum membrane. And the SRP will go and associate with the SRP receptor. Make sense? Well, next to the SRP receptor, we have the protein called a translocon right here. So translocon. And once we have associated with the SRP and SRP receptor, they will hydrolyze their GTP and it will become GDP. The SRP falls off. Bye-bye SRP. Once SRP falls off, we can start making our protein again. In this protein, the way it enters the ER when it's being made is through the translocon, which is being shown here. So our protein's going through into the ER. This signal that initially caused the SRP to bind to us, we really don't need it for the most part after we're in the SRP. So we usually chop it off. That's not always the case, but a lot of the times we'll chop off the signal peptide. Inside the ER, we have a chaperone, HSP70, heat shock protein 70. This chaperone helps to fold our protein inside the ER. Inside the ER, we will go through post-translational modifications like in carbohydrates. And then we'll continue to fold. If we are a membrane protein, we will have a part of us put in the membrane. This is membrane proteins only. This part. If we're just going to be secreted, we don't get buried in the membrane, right? So if we're secreted, we're not in the membrane. If we are an integral membrane protein, this is how we are put in the membrane. This is how we get our integral membrane proteins into the plasma membrane. We don't insert them into the plasma membrane at the plasma membrane. We insert them into the ER membrane. We bring that section of the ER membrane over to the plasma membrane and put the whole thing in. And that's why a lot of times we need flipase and flopase as well, because the lipids in the ER membrane aren't going to match up to how they are in the plasma membrane, and flipase and flopase will make sure that is all correct. And we will have a new integral membrane protein. All right, I know that's a lot of information on one little slide, but did I lose anybody? Or does that make some sense? I know you probably have to look at it a little bit more. Back through, I'll give you the 45-second version then. If a protein has an N-terminus signal on it, SRP will go and bind that protein while it's being made, step one. Step two, GTP binds on the SRP, stops synthesis. That's step two. Step three, SRP goes to the SRP receptor. Step four, GTP is hydrolyzed to GDP. SRP dissociates. We continue protein synthesis. We cut off our signal peptide. We fold our protein. We add post-translational modifications. If we are a membrane protein, we'll put part of ourselves into the membrane. If we're not, we will not do that. Why inhibit synthesis if you're just going to initiate it again? Because these proteins need to be secreted or put in the membrane if they are made in the cytoplasm. So this is cytoplasm down here. If they're made in the cytoplasm, that's not going to happen. right? Because they have to go to the ER and they need to go to the Golgi apparatus, which we'll talk about on Wednesday, to be secreted. So you have to take them to that place for that to happen. That's why you have to stop, because if you let it continue, you're just going to make a protein at the cytoplasm. Then once you're at the translocon, where you can just shove your protein through that little pipe, you're ready to go again. What does the signal peptide do if it gets cut off? That's what it does. It's like a shipping tag where it's saying this protein needs to go to the ER. So the very first thing you make is a shipping tag that says, take me to the ER. And the protein that recognizes this shipping tag is called SRP. That's what's going on here. SRP is like the FedEx truck. The signal peptide is your shipping, it's like your shipping address form. And GTP and the SRP receptor is more or less the house you're delivering to. And the translocon is the front door. And the ER is the inside of the house where you unbox your package and build it. I guess there's a good analogy. Sometimes you throw away the shipping tag, sometimes you don't. Sometimes what happens is that you put that signal peptide and it goes right into the membrane. So it depends on what protein you're talking about. Thank you. I just came up with it. I feel very proud of myself. Let's see how much. Okay, we have just 3 questions to go. I think I have the answers up already, but I'll just go through these really quick because we are done with class. So let me just give you these because this is going to be, so the SRP are the secretory pathway that we just talked about. That is the last material on the test. So a question, let's say we have a mutation in the signal peptidase. So the signal peptidase, that's what cuts the N-terminus signal. And let's say there's a mutation so that it will only ever cleave between 2 Lu residues. What might happen to your protein? So the effect should be 2 effects. 1, it can go into the ER no problem. Because the signal peptidase, if we go back to our previous slide, it only cuts after you have restarted making your protein in the ER. So we can deliver our protein to the ER no problem. But what could happen is that your protein could possibly misfold, because some proteins need that peptide cut, right? And if that peptide is not cut or cut incorrectly, you have amino acids that are there that shouldn't be there. And so you will have an misfolded protein. Or maybe naturally in your peptide, you have 2 leucines that are next to each other that are supposed to be part of the peptide, and you just kind of like cut between them. You have now destroyed your protein. So effect 1, it can still go to the ER. Effect 2, possibly misfolding. We don't know, depends on the protein. Questions about that? Okay. The blank, which is found embedded in the endoplasmic reticulum, is a blank protein. So the translocon, which is in the ER, so that's what we were just looking at, right? The translocon is like the tube in the ER membrane in which the protein goes through, right? That's protein. That's translocon. So the translocon, which is in the ER, is a transmembrane pore. So transmembrane goes to the membrane, pore because it has a hole in it. So that's C. And then question 7. So signal peptide sequence, which one is true? So that should be, I have 1, 2, and 4. So D. So let's take a look at this. It's often used to direct proteins to the lysosome for incorporation into the lysosome. Okay, so we didn't talk about that use, but not only do we have signals that go to the ER, we also have signals that go to the lysosome. Is usually not used for small eukaryotic proteins. Again, a lot of these we didn't talk about, so this is kind of a crap question now that I look at it. Yeah, so if it's really small, you usually don't have like an N-terminus signal peptide tag for it. Usually cleaved off. Yes, it's usually cleaved off. So looking at this, we didn't talk about like half the things. So don't worry about question 7. I'm not going to ask you about the lysosome or small eukaryotic proteins of that. You should probably know reason 4 though. All right. Finally, that ends all of the information for test 3 on Friday. Remember, we do not have a reading guide for Wednesday. But any questions? Can you put up the answer? Yeah, I should be able to do that right, after I get done with this. They should be online, just hidden. Will there be any math problems? You really don't like math, do you? I don't think so. I have to look back at all the material. I don't know if we did any math problems this section. I'm not going to pinky swear. I make no promises. I haven't made the test. Like I said, I'm going to make the test on Wednesday. Or I'll probably make the test on Tuesday, while our campus is being swarmed by possibly people not wearing masks, if the email is to be believed. Are the points going to be similar to the first or second test? How are the points different between the first and second test? Like, is the first test, I just left everything in, like, the first one was like 300 points, and then test 2, I made like 50 points. If that's the case, more like test 2. But the ratios are usually the same for the most part. Like, if there's a question worth 10 points on test 1, then there could be another question worth 30 points, while on test 2, if there's a question worth 2 points, there could be one worth 6. I guess I'll be more like test 2. Honestly, that shouldn't matter. I can make the test out of 1,000,000 points as long as I scale everything. So I might just do that for fun one day. Make one question 1,000 points and make another question 5,000 points. But no, I probably won't do that. Probably be similar to, I think I might do that for Gen Chem 1 next semester. Now that I think about it, test 1, make my first test worth 100,000 points. Watch them panic as they get one question wrong and lose like 5,000 points. All right, that's enough of me rambling. If you have questions, please do email me. Otherwise, take care, everybody. Make sure to go vote and all that on Wednesday.